Okay, so next up, we're going to have our specialist, Lance, who I'm sure needs no introduction from most of us here, who's really going to be exploring those capabilities of Sample Manager Professional, looking at sort of tips and best practices to help you maximize the value of that subscription, especially again in sort of these turbulent times. We want to make sure that people are getting the most out of the software that they're paying for. So. With that, Lance, I'll stop my share and let you take over.
All right. Well, thank you so much, everyone, for joining. For those of you who don't know me, my name is Lance. I'm a product specialist here at LabKey. I help prospects evaluate Sample Manager for their specific use case, and then I help onboard and serve as the customer support representative for our Sample Manager clients. Before LabKey, I worked in the lab as a lab tech in organ and tissue donation, and I've also worked at a zoo to help with the breeding of critically endangered species that would then be released into the wild. So I've worked on everything from spreadsheets to paper records and different limbs to help kind of track my data through all of these different organizations. So I'm really excited to share how Sample Manager can help because it really does tackle the challenges of these different systems and helps to improve your lab's processes. So today we're going to be discussing the professional tier of Sample Manager and how you can get the most out of its many features to help further centralize your data. And in turn, this helps to make it more queryable to find the data in the samples that you need. So as a basic overview of Sample Manager or of LabKey and our products, we have Sample Manager Starter, which focuses on that freezer and sample and inventory management. Going to professional, you'll get everything in Starter, plus workflow management, assay data management, and ELN and API access. Moving towards LabKey LIMS, we're going from a more manual process of Sample Manager to more automation to help improve your workflow efficiency. We also add visualization tools, so you'll have like really cool charts and graphs, and then plate management. Then with LabKey LIMS Enterprise, we have a lot more integrations and developer tools. So you can kind of integrate directly with our reports. So these really help to stack on top of each other from one tier to the next to make sure that you're paying for what you actually need. And that way we're also a scalable solution as your lab grows. So if you're a startup lab, you hope and then you will grow, we can help grow with you. So with that basic overview, this is a Sample Manager user conference. So, let's really focus on Sample Manager Professional. So we're going to go over an example workflow that kind of goes over all of the new features from Sample Manager Professional and kind of how it can help your laboratory build on that. So with Sample Manager Professional, we'll start by selecting which folder we want to look at. This is how we're partitioning our data to kind of control who has access to what. Then we'll use our API to import our data from a third-party system. From importing our samples and our sources, we can then add these samples to our workflow to track what steps needs to be carried out and who needs to do them. We'll then import our assay results for these specific samples. Then using calculated columns, we can verify that our results are within our expected ranges. And we'll kind of track our experiments and fill out important forms with our ELN. And then from there, you can use Sample Manager to search for your data, export it through our API, and do whatever else it is that you need to do with them. So in Sample Manager, we partition our data through folders. This helps to further keep our samples and sources separate from each other, and really specifies which users have access to these specific samples and data groups. Typically, organizations will use folders for each department or study they have and partition their data in this way. This allows groups that share some spaces like freezers to collaborate but still remain separate. So in this video, you can see that I have a bunch of different folders for different projects. Each project has different sources and different sample types available for them to use and have access to. And then if you're kind of going through here, you can see I have access to all these projects, but if certain users don't have access to Project Alpha or Project Zeta, they wouldn't be able to see these and those dropdowns. To control these and what sample types are there, source types, you can go to your admin settings and check or uncheck what each folder should have. You can also do this under a specific source type and sample type and checking which projects that sample type should be visible to. You can also archive your folders, which is a feature new from last year. If that study or project no longer exists, but you still need to see that data, you can archive it, and it makes it a little bit more user-friendly and easier to kind of view everything, and just makes it a bit more clean because we're kind of hiding them under that specific dropdown. You can also control what roles each user has for each folder. They don't have to have the same roles. So if they're an editor in some folders, they can be a reader in others and not be able to change things, but they can still view it. Alternatively, folders can also inherit permissions from the home folder. If you just want everyone to have access to them, it could make it a little bit easier. So moving on from our folders, now let's kind of import data with our API. Our API can help programmatically perform routine and automated tasks like importing that data, updating it, checking samples into storage, and adding assay data. We use languages like Python, RScript, and Java to help make sure we can communicate with other third-party software. So some folks will import their data from LIMS or a management system like REDCap or other databases, and this helps them to streamline and automate that import process of Sample Manager and reduce the risk of importing data incorrectly. Every time you're going to be retyping your data, you always introduce that risk of having a typo. I know I make typos all the time, so it's going to happen. No one's perfect. We can also use our API to query and manipulate data in a repeatable and consistent way. Like if I need to export all my samples that I add every day through a script to send to a client, I can do that. Or if I'm looking for samples that were added with specific values, maybe I'm looking for samples that need to be retested because they're outside of range, I can also do that and export that list of samples. You can also use this API to send the data in Sample Manager to a third-party tool to analyze and visualize your data. This is a bit more integrated in LabKey SDMS, but you could potentially use our reports to help with this process, make those visualizations to kind of get those kind of analysis done. We do have some developer documentation if you want to learn more about the API and see examples of what it could look like for your specific lab. So feel free to reach out to us through that help email. We'd be more than happy to kind of work with you to get that going. Moving on from our API, once all of our samples are added into Sample Manager, either manually through that file upload like those spreadsheets or through our API, we can then add those samples to our workflow. Our workflows can be a step-by-step process, typically like an SOP, to make sure users know exactly what steps they need to carry out, what samples they need to work on, and when those tasks will be due by. You can also enter data that's relevant to that workflow. Like if I'm adding samples to a shipping workflow, I can enter in the courier and the tracking number to be associated with those samples. And in case those samples get lost, I can go to either directly to those samples or to that workflow job and find that tracking number in order to contact that corresponding courier to see what's up. So in this video, you can see that I'm selecting samples that I want to work on. I'm going to be starting a new workflow job and adding them to a templated workflow. Once I do that, I'll enter in my job information, it's like my name of my job, notifying what groups or users need to kind of have updates on that specific job, who's going to be assigned to it, and whatnot. Once they're assigned to those workflow jobs, they'll receive an email, and then they'll also have a notification kind of on their homepage that, hey, we have these jobs pending being done. You can also set in those due dates so they know exactly when they should be completed by. From here, I can also assign specific tasks to individual users or groups in case some people can perform certain tasks and others can't. Each task can also have unique due dates, so you know when things need to be completed by to help with that project management piece. Um, and I did say you can assign tasks to groups. That's also a new feature, so you might see that here. We also have made it easier than ever to search for samples under the workflow UI to look for more samples to add to the workflow. It looks more similar to the sample grids, so you might see that this page looks a bit different than when you've previously used it. So I think that's kind of a great enhancement for Sample Manager. Once my workflow is assigned, I can go through my tasks, mark them as complete, and move on to the next step. I can check what samples I should be working on, quickly see where they are in storage, and kind of pull those from my freezers. All of these tasks that you see laid out here can be as specific or as generally as you need it. Like Everything else with Sample Manager, it's very configurable to meet your needs. So I know when I was working in the lab, we worked with a bunch of different products, but each product had to have specific buffers or reagents added to it. And we were working with hundreds of products, so it was very difficult to keep track of what solutions we need to be using. Some mistakes were made, but using this workflow, I could detail the exact buffer or exact reagent that I need to be working on to help reduce those mistakes and kind of make sure we're keeping up with the quality standards we needed. Um, Kind of moving on from workflow to our assays. In Sample Manager Professional, you can further centralize your data by tracking your assays or test results. So you don't have to use other systems. It can all be in one central location. These test results will be linked to the samples that were tested, as well as any parent samples or sources that sample is linked to. So this means that you'll be able to view your test results that are under a specific study or a specific patient very easily. And that way you can kind of see any patterns that might be happening with those tests. With the data being connected, you can also easily find all samples with specific test results or ranges through Sample Finder. So again, we make it very easy to search for the things that you need to get the most out of your data. In this video, you can see we pick up kind of where we last off with our workflow. From our workflow, we can import our assay data for the samples in the specific job, so we know exactly which samples are going to be tested. I can then manually enter in my run information. In our run information, it's what's going to be the same for all of our samples. Usually this is something like a test kit, testing instrument, or an operator. I can then manually import my assay results, import those results through a spreadsheet that was spit out from a testing instrument, or if I'm kind of moving things around in Excel. Or I can use my API to connect to a third party and add that assay data into it. It's very similar to adding samples, so there's really not that much of a learning curve to add this new functionality. Once I kind of add all my data for my assays, I can filter through these results and see how they're connected to my samples and my sources. This helps me to find any patterns and outliers that may need to be addressed. So here is kind of like our sample grid. All of these columns are filterable and easy to kind of go through. I can also kind of click on that sample, go to that assays tab, see what those results look like. If they're going to be changing from assay to assay, I can also then go to my lineage, click on my mouse or my specific patient or study, and see all of the results that have happened under that specific patient or mouse. So this again can help find those changes or that you kind of want to filter out. When I go to Sample Finder, this is also where I can find results that may be missing, where if I want to find all my samples without analytes results, I can very quickly do that and pull up a list of things I might need to look out because we're very busy in the lab, mistakes happen. So we want to try to find those issues when they come up very quickly. New to Sample Manager as of late last year are our calculated columns. So our calculated columns are very powerful and can help with a wide range of things from calculating data in a grid for automatic results, flagging values outside of an expected range, and autofilling cells based on different selections. So we'll kind of go into more examples later, but some examples can be that if my pH value is greater than 5 or less than 2, I need to set a flag for an invalid result, right? It should be 3 or 4, I guess, for the specific experiment. I can also use it to calculate my dilution of my reagent based on the volume of what I'm using. Or if any consent fields for my samples are marked as no, I should fill in no test so I know not to touch it. So these are very basic examples, but it just kind of shows we can do quite a few things with these different fields. So in this video, I'm kind of showing my sample grid that has a couple of different calculated columns. I have a column that calculates a tube discard date to be 7 days after the draw date. And then I can also see that there's tube errors to catch any discrepancies. So here, if I'm entering a tube type that's an SST or a red top, I would need to mark that as a serum. But if I mark it as plasma, it should give me an error. And then if I mark it as an EDTA, it should be marked as plasma. And if I mark it as serum, it should have an error. Very basic example, but it kind of gives the point that we can kind of catch any discrepancies there. So my calculated column tube type error serves as an easy way for me to catch those and flag me for it to fix. I can filter through those columns for, hey, there's an error here, and then update it. When I fix it, that calculated column will automatically change to get rid of that error message. You can add calculated columns kind of when you're editing your sample type designs, your source type designs, your assay designs. So they're in a bunch of different places. The sample manager doesn't have to just be in one location. So here I'm just kind of adding samples to kind of show what those calculated columns look like when you're entering in that data. And this is where I'm going in to edit those calculated fields. And this is just kind of like an example formula of what I use to kind of catch those errors. So it could be something really complex like that, or it could be pretty simple where we're just adding on a week from a date. Or it could just be we're adding one grid or data column to the next. We do have example documentation on what we can use. So we have LabKey SQL where it gets a bit more complicated. It has a bunch of different functionality, which can definitely be overwhelming, but it's great that we support a lot of those things. And then if we kind of go to our assays, I kind of pull up another example where it's a bit more straightforward, where it's like we do adding and subtracting and dividing. Anytime you're doing a manual process here in the lab, it introduces a new way for you to have an error. So us being able to automatically do that for you will hopefully save you from trying to correct a bunch of issues. So if you have any questions on those calculated columns, feel free to email us, let us know. We can kind of work with you in getting those set up to match what kind of calculations you want to do. Um, and then I do have in my next slide some example cases. So in this chart, we have different calculation columns and their formulas. We can calculate new dates. So this would be if I want to know when something will expire, when we should discard it of a sample, or check on it for testing. We can always insert a line of text to make reporting easier when you're exporting your grids. So this is like, it should always have the word added in these data columns. This is usually if you're exporting your data into a spreadsheet for a reporting in another software, or maybe you're making mailing lists for templated results and you don't want to manually update those spreadsheets, you can just have Sample Manager add those columns for you right away. We can also display a new value if something else is selected. So that was kind of the check of I select SST and serum into columns. I should just ignore it. It's not an error. But if I select SST and plasma, I need to have discrepant data or another error message kind of pop up. And then we can also truly act as a calculator where we're calculating new values based off of other things in that grid. So that's kind of where I'm calculating a new concentration for a solution through adding, subtracting, multiplying, and dividing. And then these are kind of just example formulas, but we can work with you very closely to make sure we get those formulas right for your specific use case. Once we're done with our experiments, and maybe while we're doing them, we can track our notes in our ELN or our electronic laboratory notebooks. So we can use our ELNs to replace paper records or forms through kind of templating them in Sample Manager. This kind of helps reduce legibility issues because I know wearing all that PPE, you're in a rush. Sometimes your handwriting just isn't that great. It can kind of help with those issues that we see quite a bit working in the lab. These paper forms and records don't have an audit trail. So if someone doesn't write in or fill something in, it's sometimes really hard to tell who did what. But with our ELNs, we have an audit log, and we also support electronic signatures to further support compliance for that part 11 compliance. This helps you also collaborate others very closely. So if I'm working with my quality department and filling in audit checklists, I don't have to walk all the way over to them, or working remote, I don't have to email them. It's all going to be in Sample Manager where we can all log in, work on it together. Doesn't matter where we're at. It's very similar to Sample Manager where updates in real time. And these can also help track your steps in more detail and help others replicate your work to support the scientific process, right? So with research and with new experiments, we always want to make sure that we can replicate it to find discrepancies in our experiments, find ways to improve our understanding of the world. And like I said, with other data in Sample Manager, ELNs can be linked back to samples, so you can see where they're being referenced, better access important information, and further centralize your data into one location. So in this video, you can see I have a list of my recently opened notebooks. I can then look more at all of these notebooks, filter through them to see what I really need. So if I'm looking for all my notebooks from a specific template I set up, I can do that, or look for specific authors, time ranges of when we've created them, or their different statuses to really help get to the exact notebook we need. I can create new notebooks and collaborate with other authors. So this is where I'm selecting a specific template that I've used, entering in notebook titles, and that way, if other authors are in here, they can also edit it with me. This is great if we're, working on the same experiment together. So here I just kind of selected my audit checklist, and then I'm going to create this notebook. These notebooks can have different sections, so I can quickly jump from one section to the next. So if I need to look at waste management, I can quickly jump there. This is great if your notebook has a lot of entries. Sometimes people will use specific days that they're entering in the notebook as an entry, so you can jump to the specific date that you need. With these notebooks, you can also tag specific samples, sources, assays, workflows. It's very similar to social media where you type in that @ sign and then you tag whatever sample you need. So it's very easy for new users to kind of pick up and learn. These notebooks can also have checkboxes, inline images, and tables. So for this specific experiment, we're tracking things in our audit checklist. So if I need to mark, is the laboratory well maintained? Yes. Or if it's no, I can also mark the threat level of that compliance issue. Once I'm done with this notebook, I can send it off for an electronic signature. I can select the members that need to sign off on it or the group. And then they would log in, verify their login credentials, so their email and password to help with that compliance piece. If it's all approved, they'll sign off on it. If they need to send it back, they can definitely send it back for review. Or if you submit it to your manager, like, oh, there's a typo or I forgot something, I can also recall it, make those changes, and then resend it for that signature again. So with that, this was just an example workflow process of how you can use Sample Manager's professional features with our folders, API, workflow, assays, calculated columns, and our ELN. This is, of course, very configurable to meet your specific lab's needs, and we'd be more than happy to meet with your lab and help get these set up to further centralize your data and get the most out of our software, because we think if you're using multiple systems, it can also help save money, but it can also just make sure you're kind of keeping those workflow processes more streamlined and cohesive with one another. So thank you all so much for joining us. I'll be here a little bit longer to help answer your questions. And of course, should you need more one-on-one time to chat, or if you think of some questions later, please feel free to email us at help@labkey.com. And I hope you enjoy the rest of the Sample Manager User Conference. It was great hearing from Wendy at Gen QA. I'm excited to hear from Camilla with Alaska, and Steve later on for our data integrity. So let me know if you have any questions or if you want to dive deeper on any of these features. We can look at how to get these different features set up if that's something that you'd like. But thank you so much.
Thanks, Lance. I think we'll give people a couple minutes to sort of add their questions in the Q&A. In the meantime, I think what would be really interesting is talking about some of the different scenarios that people have used ELN, even sort of deviating from that traditional electronic lab notebook. One that comes to mind is a customer that is using that as a replacement of their inventory checklist that, traditionally was on a piece of paper that they would sort of check off that they checked inventory. I know that they're now using that ELN functionality to basically have it all centralized in the same place. Are there any other scenarios you want to share for folks?
Um, I know people will just kind of have it as like a to-do list of them to kind of check things off. If they don't want to have like an ad hoc workflow, they just want to edit that workflow or the type checklist, I guess, as they're going, they'll do that. And like audit checklists or just other forms where they're kind of tracking courier information, that's another place that they can do that there.
Yeah. I was also thinking like potentially if Wendy was interested in sort of adopting that feature, there could be a way to sort of put all the files associated with a given sample sort of in an ELN as well, which would be potentially quite a bit easier to manage and export and stuff like that. So great. Okay, looks like we've got a question here. So in certain storage locations, we want to prevent storing aliquots of the same sample together. Is it possible to have a pop-up notification when adding a sample to these locations, um, kind of notifying that there's already a sample present there? Any I can maybe speak to this a little bit. We definitely don't have this functionality sort of built out. I haven't heard of this scenario before, but I can imagine it's sort of as a way to prevent some redundancy, or to have some redundancy, not prevent some redundancy. So I think that's certainly interesting, as a potential thing for us to consider what to do. As mentioned, kind of in a different Q&A while Lance was presenting, that type of business logic is stuff that we're really trying to build out into our workflow functionality in LIMS this year. So certainly a good scenario for us to consider.
And one thing I would do with that is for if I want to have like my parent samples and my aliquots separate, I would use my box labels to have these are for my parent samples, these are for my aliquot samples, just to kind of help with that process.
Yeah, that's a good point. Um, and then, okay, how many options can you add when using a conditional calculation based on a text match? For example, the field properties reference shows a case when color field is blue, then abnormal, else normal. But what if the color field is red? Can I create another option within that expression?
Yes. So I haven't ran into a limit just yet. I'm still experimenting with it. So you could definitely do that, which is fantastic. The formula is going to be quite a bit longer, but we should be able to support it.
Yeah, and I do think that we've built out a pretty large sort of character limit in that SQL expression as well. So that's much more likely what you'll sort of run up against is how many characters can you sort of type into that box effectively that we save. So, any other questions for Lance? Are we able to reference parts of the notebook information or custom fields under the sample tables if the sample is referenced within the notebook? Parts.
So is that like the metadata
Yeah,
for
I'm not sure. Reference parts of the notebook information. Um, Oh, so I think maybe Jill, are you asking like to be able to pull in data about the ELN from within a grid view? Um, no, that's actually not something that we've hooked up yet, but I could imagine, yeah, having that utility, being able to find things. One of the things that we do want to sort of build out more is the ability to find samples based on what workflows they were part of. I could imagine doing the same for Sample Finder with like what ELN templates or what metadata kind of captured in that ELN is another really good feature for us to think about. Awesome. Okay. Thanks, Lance.